Hi everyone, this talk is about the generation of a human cervical cord template from high-resolution data. I have nothing to disclose. MRI templates provide a reproducible and standardized way to process and analyze large amounts of data. Spinal cord templates have been successfully developed in recent years. The resolution of these in vivo templates are however limited to 0.5 mm isotropic, which compares a correspondence with histological descriptions of the anatomical spinal cord structure at microscopic resolution. The goal of this study was to generate a 3D human cervical cord template at 80 micrometer isotropic resolution. From ex vivo data, this template is based on C2 to C8 spinal levels. Data acquisition was performed on 13 subjects, males and females, average age of 76 years old. The same extraction and acquisition protocol have been used for all subjects. It has been fully described by Asiri and collaborators in 2016. In particular, we can see on the photo on the right that the nerves have been carefully preserved during specimen extraction. In this talk, I will focus on the template generation from this data. Methods. First, data labeling. The spinal cord segments were identified by labeling the rostral and caudal extent of each nerve root. For the tissue segmentation, we trained a deep learning model using the open source framework Ivadomed, which allowed us to accurately and efficiently segment the gray and white matter on around 1,000 images for each subject. Second step, we followed the same process as for the PAM50 template generation. So spinal cord straightening, then spinal level alignment across subjects. These operations are implemented in the spinal cord toolbox. We also applied some bias field correction, intensity standardization along the inferior to superior axis. Template generation. It was performed using the animal framework, using iterative nonlinear deformations. In particular, the process is unbiased, which means it doesn't rely on the selection of a single subject as a reference, and the template is right-left symmetric. Once the template generated, we brought the spinal level labels in the template space, generated a probabilistic map of the gray matter based on each subject gray matter segmentation, and the motor track labels that have been manually segmented directly on the template. Here, some axial slices of non-processed images from different subjects, and at the bottom, the result from the automatic segmentation for the gray and the white matter. Now, template slices from C2 to C8 spinal segments. In particular, we can observe a fine delineation of the gray matter anatomical structure. For instance, the distinct regions containing motor neuron pools that control the axial muscle, deltoid and bicep at C5, and tricep muscles at C7. It is even more clear when looking at the probabilistic maps of the gray matter, from dark blue to yellow, which visually corroborates with histological studies. Motor tracks labels are also available along the cervical cord from C2 to C8 spinal segment. We believe this template and atlas could facilitate large and robust study of the association between function and structure in the spinal cord. In particular, it could be used for fMRI experiments since it is based on spinal levels. Limitations of the study include coverage limited to C2 to C8 spinal levels, a null population used to construct the template, and tissue shrinkage that can occur during postmortem fixation. We have made all the material of this project open source. This includes the processing pipeline, the deep learning model for segmentation, the template, the gray matter and motor track atlases. Also, the template was registered to the existing in vivo PanFIFI template to enable further atlas-based analysis with high-resolution data. All this is available in the GitHub repository indicated in the yellow box. Do not hesitate to open an issue there if you have any questions. That's the end of the presentation. I would like to warmly thank my colleagues from both Brisbane, Australia and Montreal, Canada for their precious support, and you for listening to this video. Thank you.